Okay, hello everyone. Okay, so I'm Dr. Irina Savina from University of Brighton, and I will present today my talk about macroporous cryogels and their application for biomedical and environmental field. So on the bottom, you see this nice, I don't know if you see my cursor, nice pictures. They're all cryogels. So before I will start talking about cryogels, I just want to give you understanding how they're different from other hydrogels. Basically, cryogels is hydrogel, but the way how we make them, it's by freezing before they're making the gels. They have completely different structure. So if you look on these two standard electron microscopy images, so this is the dextran cryogels, which was made from the same solution, but one was done at room temperature on the right, and then the one on the left has been prepared at -20 degrees. So you see there's dramatical difference in morphology of these gels. So the cryogels has macroporous structure, and you see that it has dense polymer walls. And because of that, mechanically, it's also different. The gels which is prepared at room temperature will be more brittle, and the cryogels is sponge-like, and mechanically is normally quite strong. It can be compressed significantly before it starts to break. So that is a picture that was done by one of our colleagues a long time ago, but I think they kind of capture that considerable difference between cryogels and hydrogels. So how the structure is made? So this is very simple physiomechanical approach. Here we just take the initial solution. It could be any solution which can make hydrogels. It could be contain some monomers, an initiator for polymerization, or it could contain polymer and then cross-linker. And then what we do, we just freeze that solution at normally temperatures about -12, -20 degrees. So what's happening then is a solvent, the water, will be freezing out, forming ice crystals. And then because we freeze before the polymerization happens, the polymerization will happen in a small region between ice crystals, which we can call that as a small interface, which remains non-frozen. And in that kind of interface, it's all our monomers and polymers will be concentrated. And then when polymerization has happened, and when we defrost the sample, what we will have in a place where we have ice crystals, we will have voids filled with water. And then the polymer which is formed will be quite dense, because all precursors was concentrated in that small regions. And then we have the structure which you see on the bottom is a very highly macroporous gel. So the interest to that gels is raised since the beginning of the century, and mainly it's what is attracted in the beginning is that it has big macropores, and that big macropores, if you see on that image, is between 10 to 100 micrometers. So they quite good size to accommodate cells, for example, or some microparticles. And also, these pores are interconnected, so the materials become permeable. And this is all these properties attract the attention of the researchers. So the macroporous, the permeable, the mechanical properties depends on the composition of the polymer or what additional materials you will add, like nanoparticles will affect mechanical properties as well. So in the way you can tune the properties, it depends on how you will use it later in applications. And then you can make them in polymers from synthetic or from the biological-based polymers, and then they could be biocompatible or be less toxic and biodegradable. So you see it's on the images as well. It showed the compression. So if we compress the gels, and then it's you see that the water is simply just squeezed out from the gel. When we release the load, we see that the gels is taking the same shape as it was in the beginning, and just basically we have repeated a few times and see that there's not much damage has been done, depending on what conditions you made the gels and what polymer you use. It's also easy to reswell from dry polymer. So all of this is then leads to different applications. So the first application was a bioseparation, where there's a group in Lund University where started working with macroporous gels, was interested in using them as in a chromatographic column, monolithic chromatographic column, to separate different bioparticles and even the cells. And we also can use them as a filters, for example, to separate proteins from the very complex mixture, which can contain cells, debris, and other microparticles. Then it's also attracted attention tissue engineering, as a cartridges for tissue engineering. And the first application was skin substitutes for wound healing. And then there's other also application is to using them as artificial liver support for bioreactors, or even in there was tested to be used in high throughput screening. So we're coming to the wound healing applications. The interest to that materials was it's basically to support the healing of the severe wounds. When you have the burns, which is very extensive, you need something to cover it very quickly, and you also want to stimulate that wound healing processes as well. And there's different materials has been studied for wound healing applications, and mainly it was focusing on biocompatible polymers. So in the image here you see, and this is gelatin-based hydrogels, cryogels. And then this is scanning electron microscopy, which is quite clearly show you the green is a polymer stained. And then you see blue dots, it's a nucleus of the dermal fibroblast, which was cultured inside of this material. So you can clearly see the distribution of the cells inside of the cryogel material. So the first, what we want to show that is actually cryogels is could be developed in the way that is we can compare it with material which exists already on the market. And the golden material we took, the Integra. And Integra is collagen-based hydrogels, and then is because it's a collagen, it's basically quite expensive. So we come with ideas. It's that to use something cheaper as a gelatin, and but we also need to analyze and understand if that will support the cell's growth and migration inside of the matrix. So on that work, which is done by my colleague Shevchenko, he shows that is A is Integra, and then B is the other way around. A is the gelatin gels, and B is Integra. And you see the formation of the outer layer of the skin keratinocytes. So basically, what is in this work was demonstrated that they're very good substitute for the wound healing, and they supported the cells migration and formation of the new skin construct. In our recent work, together with a group from Kazan University, what we are looking for on how adding some metal ions to the cryogels can then affect their performance also in wound healing. So there's different ions has been selected, so the copper, cobalt, and zinc, and then there's effect on the basically on the cells. Proliferation was analyzed, and the first, of course, it was a screening when you just want to see how much metal you can introduce, because all the metals could be at some point it could be beneficial because they have been part of some enzymes and contributing to different regenerative process in the wound, but they also can be quite toxic. So from screening in the first place, it was found that the concentration below 1 mole is probably millimole will be beneficial. And then it's also different ions has a different effect. And what you see on the below image, the effect of metal ions on proliferation of ovic cells cultured on the cryogels, and you see that the copper is has and cobalt stimulate proliferation, and angiogenesis, and zinc influence collagen position and immune response in modulation of the wound. Well, then my colleagues has done even more thorough investigation studying the effect of the metal ions on the secretion of different cytokines and growth factors, and that is, you see, it's presented in very complex chart. There's different cytokines and growth factors have been analyzed. And in general, the secretion of FGF2 and FEGF and IL-6 and IL-8 was profoundly stimulated by copper mainly, followed by cobalt and zinc. So in another work, it's here the cryogels has been tested for neural tissue replacement. And the materials which we was working on is it was selected that oligopolyethylene glycol furamate and polyethylene glycol diacrylate as a cross-linker, and then also to add a little bit of the cationic monomer MyAttack was beneficial as well. So the composition was selected based on the previous research, which showed that this kind of hydrogels has a potential for developing neural tissue replacement. But in this work, we want to see if the macroporosity, which is cryogels, will have some additional effect. So the first is for synthesis of the cryogels. It's different compositions has been tested and analyzed, and mechanical properties were tested to select what will be the best composition and what will be the best concentration of MyAttack monomer need to be added. And then, so here on this slide, you see the rheological properties of that gels, which contains different concentration of PF, 2% and 4%. And you see considerable difference. The higher concentration you have, the better mechanical properties you have, more stable gels you will have. And then it's also, you see when we add additional monomer, cationic monomer, we don't see a profound decreasing in the mechanical properties. So this has been selected then for further study to analyze the cell behavior in the cryogels, and we select PC12 neuronal cells. And you see on the left-hand side, this is imaging showing how the migration of the cells is depends on if you have introduced an additional monomer, cationic monomer or not. So OPF8 doesn't has MyAttack, and OPF13 has one, and you see that there's the blue dots is actually the cells, and you see that it's considerably increased migration of the cells inside of the gel. And then the figure is showing you quite clearly that the concentration of MyAttack was important, because with the concentration we see increasing the depth of migration of the cells up to 0.8 when you have a percentage of MyAttack, and then it slightly drop. So we select the gels with 0.8% for the further study, and that is continuous work. So on this slide, you see that the cells behavior in the cryogels, adhesion and proliferation, was analyzed, and two different types of cells has been analyzed, PC12 and CHCY5Y. And you can see clearly that adherence of the cells will be depending on that cationic monomer presenting in the cryogel, as well as the proliferation of the cells. And then below you see the images which is showing you the number of the cells, which can be seen by simply just staining with crescent violet optical microscope. So in other parts of application where the cryogels has been tested is environmental applications. And first, what we looked for how to use the cryogels is just to consider to use nanoparticles for the contamination of the contaminants in water or in the soil. So nanoparticles is quite effective to the contamination of pollutants, but the problem with nanoparticles is we don't know much about toxicology, and we don't know much how the nanoparticles, when they're released to environment, where they end up, and then is what kind of the dangers can be presented to the environment. So it's in our work, we decided to contain them in a cryogel macroporous system. And by this, what we do is that the nanoparticles is now contained in the side of the polymer, but cryogels providing that permeability, and we can design the filters based on the cryogels. So we simply can filter the water through the cryogels which contain nanoparticles. And the first analysis, what we have done, we use arsenic-3 for absorption on iron-based nanoparticles. And we have done some optimistic results. And what we also have been observed that nanoparticles performance mainly will depend on the surface area. In our case, it's when we mix it with polymers and introduce it inside of the polymers, we see quite a lot of agglomeration, and that is reduce the surface area and capability. So the further work is has been done to analyze it, and this image is showing you this agglomerates is formed inside of the particles, and the flow through the composites that is going through the polymers, polymer walls, and that absorption happens in nanoparticles. So to avoid that agglomeration, so what we have done next is we actually produce iron oxide salt gel. And then it's this is a picture of that gel, which is you see have some tiny particles, but then it's we put that gel inside of the cryogels, creating macroporous structure, and we have seen very good improvement of distribution nanoparticles. We also eliminate that another drawback which happened when we tried to mix nanoparticles, which is have high density with polymer solution. We have deposition of nanoparticles during the cryogel formation, and then uneven distribution is inside of the gel. For using this method, we actually avoid that, and we have even distribution of nanoparticles inside of the cryogel. And then this lead is to much higher capacity for the absorption of the metals, as arsenic-3 tested here. So we have tested different polymers, different composition, and you see that we have seen a remarkable difference. So it's on the bottom, you see that iron oxide nanoparticles hemopector gels is giving us capacity of 0.21 milligram per gram of swollen gel. But if we're using then the new techniques of making iron salt gel, then we increase considerably the capacity to 1.3, 1.5 milligram per gram. So studying isotherms show us that the capacity of absorption of arsenic-3 was 625,588 milligram per gram of iron for original gel and the iron gel and the cryogel composites. So we see slightly decrease in the capacity, but it's not significant. And if we compare it with other studies, so we have very good results. So previous studies reported between 8 to 175 milligram per gram. So of course, if you're using that materials as a filters for the water, and that is what they was designed for, you need to consider the toxicity of that materials. And we have done the analysis of toxicity using the cells. And as a result, they show cell viability and cell lysis or hepta-G2 cells. Exposure to that materials for 24 hours is doesn't change compared to the control. So the next is if you want to use a cryogel, you're thinking about scaling it up. And as I said before, when you're scaling up and trying to make gels with nanoparticles inside, you will have this deposition of nanoparticles in. So we was working on a technique how we can improve that deposition, and then we developed some techniques which completely improved the distribution of nanoparticles, and even when you're using the carbon, which is on the picture C and D, this is the carbon-based materials, and carbon beads was quite big. We still see the distribution of them in material. Another very interesting work had been done by Dmitry Berila. It's using cryogelation technique to make the cryogels from the whole microbial cells. So as you know, microbial cells can be used in a number of applications. In this particular one, we was interested in environmental application with microbial cells will be decontaminate the water. So the bacteria was selected which can degrade phenols and phenols derivatives, and then the cryogels was produced by mixing basically the cells and some small amount of the polymer cross-linker, and then the same techniques was used to make the cryogels. And you see that on the bottom there's a picture of the wall of the cryogels, which is made from the microbial cells glued to each other. And that gels has porous structure, as you can see on the second image. So what is interesting about that this 3D structured bioreactors was made from the live bacteria, and with very minimum of the polymer in there. So this, then we test them for the removal of phenol, cresol, and chlorophenols. And we tested different strains, bacteria strains, Pseudomonas mentacina and Rhodococcus carinus. And we also tried to mix that population to make the cryogels from the mixture of the cells. So you see that the last image is where the two cells is produced together. And the beneficial properties of these cryogels, it's that is in environment, microbes already used for the contamination of contaminants. But what is the main problem is the formation of biofilm, which is take quite a long time. In this application, what we show that we can put cells together and produce this artificial biofilm just in a couple of hours. So then we want to be sure that the cells is alive there, and we tested the cells, and this image is showing you the different gels produced with using different cross-linker and different concentration of the cross-linkers. And our cells was stained, so everything is green, showing you the live cells, and orange red is the dead cells. And then we analyze it, and you see that is on the graph that the live cells will manage to keep about 50%, 50-40% of the cells alive after this chemical cross-linking and cryogenic exposure. So then we tested degradation of the phenol, and the first, when the cells was defrosted, you see it's in time 0, they not actually respond to degrading phenol well. They need about 5 days to wake up, but then it's, you see that repeatable cycle of adding phenol to the cells. We see quite is reproducible results on degradation of phenol. Then we also has been interested. It's possible to store that cells or that cryogel made from the cells in -80 degrees, and so the cryogels was stored for 4 weeks and then defrosted, and then it's, you see from the graph that it's take a little bit longer for cells is actually to wake up and started to perform in a normal metabolic way, but then they started degrading phenols on 15 days after this. So this is show that it's possible to store that cryogels in -80 degrees for transportation or other use. So also was interested to see if the different concentration of phenol is will affect the cells, because even if the cells using it as a food, so it still could be toxic. And in this, the graphs they showed that the cells was fitted with 50, 100, 200, and 250 ppm, and they still perform the degradation in different cycles. So then the viability of the cells has been tested as well after exposure to the phenol. And this graph is showing you that how the different cells to respond, the number of the cells after the phenol degradation. And we also see CF3 and CC3, it's different cross-linkers. So cross-linker definitely has effect on the stability of these systems. In another approaches, the cryogels has been used as bioreactors, and as we say in bioreactors, is materials which can hold and contain the cells and then maintain their physiological activities. And in this research by our PhD student Banalumi, she has been looking on the bioreactor to be used for bioartificial liver. So in this case, what was interesting, you probably know that the liver disease is quite affecting quite a lot of people, and one of the kind of solution for that is just basically to use a donor liver, but then not everyone can survive until the waiting for the appropriate donor liver available. So there's needs in some kind of the materials which can perform it in kind of a certain level, the activities of the liver to reach that patients to actually to the operation which is required. So what we have done is the cryogels first was produced as monolithic column, but we also studied during that research is the flow through the monolithic column and find that it's not efficient. And the most efficient way is we'll be constructing the bioreactor, which has the disk of the cryogels spaced between each other, because it's provided better exchange of nutrients and then products of the live cells. And this is the final kind of device which has been produced and tested. And what we looked at is to analyze the performance of the cells inside of this device. We looked on albumin production and urea. And the gels is different gels has been produced to attach the cells to the cryogels. It also has been used RGD modified column, and this here you can see that is the RGD modified column, which is just monolithic. It's and then RGD column, which is produced, as I explained, by the stacking different discs of the cryogels together. So you see that performance of the final design of this bioreactor is much more better for albumin reproduction cells. Okay, so finally, I want just to thank you all my collaborators, and there was many of them, and yeah, thank you for your attention.